Hello and welcome to my 10-minute review of Chapter 25, basically looking at life on Earth. Again, what you're going to need to know from this chapter are the conditions of the early Earth, kind of what a fossil record is and how we look at that, kind of the start and the relatively eras at which things kind of came about, this idea of mass extinctions and adaptive radiation, and basically talk about how gene expression can affect macroevolution. Again, a big question, something that we don't know, that we have not done in a lab, is to determine how life evolved. Basically, what we had to have for that is an abiotic synthesis of all the organic molecules. Organic means from life, and so the organic molecules now are created by living systems. But again, those organic molecules that make up life first had to come from somewhere. Those molecules had to be joined together in what we call polymers. We had the formation of what we call protocells, cells that could actually reproduce from the abiotic synthesis of the primers in there, and so we'll look at that. And then finally, we need to have self-replicating molecules and self-replicating cells. This is a picture of the early Earth, and as we can see, there's not much oxygen there. Early prokaryotes, roughly about 3.5 billion years ago, did inhabit this area, and they used those abiotic molecules for energy and for their products. We saw the production of oxygen, and that started to accumulate in the atmosphere at roughly about 2.7 billion years ago. It was cyanobacteria and not plants who were producing this oxygen. Again, the documented history of life occurs in the fossil record. And so, again, when we go and look at old rocks, we tend to see remnants of old life forms. Again, from very weird-looking kind of things to definitely things that are recognizable today. Again, the further down that we go in this soil, so these things are laid down a little bit like layers in a cake, and the oldest rocks are the ones that are deepest, and the newest rocks are the ones that are closest to the surface. So again, we are going to see older organisms 230 million years ago way down deep, and relatively newer ones like 65 million years ago. Okay. We can also use radiometric dating. That is, radioactive molecules have a half-life. Carbon-14 can be used to look at things that are relatively recent. Other types of radiometric dating are out there, and you can take a look at that. This is something that I want you to kind of know. Again, roughly how old is the Earth? When did prokaryotes come about? Roughly about 3.5 billion years ago. When did oxygen come about? When did single-celled organisms? When did multicellular eukaryotes? And basically land animals and plants. Again, be aware that most of these, the body types that we see today came about from something that we call the Cambrian explosion. And there is very not much left from the Cambrian explosion because it exploded. No, it didn't explode. But again, these beginning organisms really didn't have much in the way of tissue that was able to fossilize. And so we did see the basic body plans of almost all organisms occurred before this fossilization occurred. But again, be aware of all these and what their differences are. Again, mammals, when we look at the DNA sequence, which we'll cover in next class, when we actually looked at parts of the skull, and so bones in the skull, we have articular, quadrates, chromosomal, dentistry. Again, we can look at kind of the history of what we mammals are, and basically we share an ancestor with the reptiles and with the birds, but again, that was a very, very long time ago, millions of years ago. Plants, fungi, and animals colonized land roughly about 500 million years ago. Be aware of that. Again, there's some people who say plants came first, some people say animals came first. We weren't there, we don't really know, but again, have an idea of what adaptations allowed animals to go on there. Again, we need to know what macroevolution is and what are the conditions. We look here at this thing, we look at roughly 250 million years ago, when it's quite a long time ago, but not much was going on. We had kind of one solid continent. About at the end of the Mesozoic, beginning of the Cenozoic, 65 million years ago, we basically have our continents there. And so be aware of how the Earth is arranged and how materials move along the crust. And this one here is talking about major mass extinctions and the diversity of life. And while we human beings are very rapidly killing everything, kind of stuff like this has happened before in the timeline of the Earth. And be aware of these mass extinctions. We had one 65 million years ago that killed off the dinosaurs. Again, 200 million years ago, 250 million years ago, 350 million years ago, 450 million years ago. And so these things do occur with relative frequency. Again, we see a loss in the number of species, and then we see it take a long time for those species numbers to come back up. The KT, Cretaceous Tertiary extinction, we think was a meteor that hit somewhere in the Yucatan Peninsula and basically wiped out most of the Earth, and definitely the dinosaurs. We mammals and the birds were able to survive. Again, that allowed for adaptive radiation. Again, we mammals were around during the dinosaur times. Again, we didn't have bears and human beings and cavemen and stuff like that, but we did have mammals. But again, they weren't able to take over because the dinosaurs were so good. However, when that meteorite struck, we hypothesize, again, those organisms weren't able to control their temperature. We survived, and so what we see is an adaptive radiation. We were there, and we were able to move into a lot of the jobs or the niches that those other organisms have. And so again, be aware of that. Again, how can we see these big changes? And we talked a lot about mutations in specific genes. Genes that make you bigger, genes that make you faster, but not all genes basically have that effect. Some genes can affect the growth pattern, and a lot of our evolution comes in changing developmental rates. And so in this one here is looking at developmental rate of the chimpanzee compared to human beings, and we see that we human beings have more of an infant-like skull, so maybe we didn't develop as much. Again, we will talk about it next semester. There are these Hox genes that control when a gene is turned on and turned off, and we see that, again, we get things like snakes because the genes that are there to turn on the limb formation don't really show up. Okay, and so we can have things also like the development plan from something that looks like a fairly simple crustacean to an insect occurs because, again, those Hox genes that turn on or turn off limb formation, in this case here, the limb formation is turned off. And so, again, we have a powerful tool for the production of new genes without really changing the type of gene, just changing the timing and changing the on-off switch of when a gene comes on or off. All right.